Hello, welcome to Science 30, Unit A, Chapter 2 on genetics. So this is the first lesson in our Chapter 2, and we are just going to have an introduction now to what is genetics and look at some of the terminology that you're going to need to have an understanding of as we move through the lesson. So take a look at your outcomes for this lesson and make sure you check out your textbook pages as well. Remember that the textbook does have a lot more extra information that's not necessarily required for you to understand everything. So I am definitely going to hit all the key pieces that you need to know in this lesson. All right, so starting off with DNA, we've all heard the term DNA. You may have even studied it before in grade 10 or 9 or even earlier. So what is DNA? It's actually just stands for deoxyribonucleic acid, DNA, and this is the genetic information that is passed down from your parents, grandparents, great-great-grandparents. This is the information that lets you know in your body know that you are human, for one thing, and codes for everything in your body, everything. So where is this DNA found? Well, DNA is found in every single cell of your body, well, almost every single cell of your body, and it is located in the nucleus of each cell. Okay, and the DNA structure itself, you can see it's this cool double ladder, twisted ladder called a double helix. There it is. And we're going to learn more. We're going to delve deeper into the structure of DNA in the next few lessons. So this is just an idea, basic overview right now. Okay, now DNA is really, really long. There's a lot of DNA in those cells. So what happens is it's coiled into this form called a chromosome. So speaking of chromosomes, let's take a look. What is a chromosome? So chromosome, like we said, is DNA coiled really, really tightly. One chromosome is going to look like that, but most of the time they hang out in duplicated pairs that are attached together at some point, and this attachment point is called the centromere. So they come in this very characteristic X shape. Okay, so these contain all your DNA. Now, the number of chromosomes that are in cells differs between organisms. So in, say, something like an ant, all ant cells have 2 chromosomes in them. Okay, it's the same chromosomes in every cell, but every cell is going to have 2. Dogs, 78 chromosomes, cats, 38, shrimp. So apparently size doesn't matter how many chromosomes you're going to have. Shrimp have 254. So humans, so your cells, if your body, have 23 pairs of chromosomes. Okay. Now chromosomes are always found in pairs. You get one from your father and one from your mother. Okay. So what's nice about chromosomes is because the DNA is so concentrated, we can take images of our chromosomes and get a good visual image of what's going on here. So when you take these chromosomes and you order them from largest pair to smallest pair, let me just go back there. Whoops. You create a karyotype. It's called a karyotype. There you go. So there's 2 karyotypes here. There's the human female, alrighty, and then there's the male karyotype. Now, my picture is in the way of something very important, so I'm going to move me over here. Excellent. Okay, so you'll see there's 23 pairs or 46 chromosomes in total if you were actually going to count them all. I'll use the male one because it's been counted for us. So there's your first pair here, your 22nd pair, and then here's your 23rd pair. Now they didn't use the number 23 to represent this because these are the sex chromosomes. These are determining the sex of the organism. So we refer to these as the X and the Y chromosome. So you'll notice that the female has 2 X chromosomes and these are the longer chromosomes. And a male has a X and a Y, and the Y is a shorter chromosome than the X. So that's what determines if you have a male or a female, that chromosome. So human karyotypes can be used to identify some genetic disorders. For example, Down syndrome is caused by having an extra chromosome 21. So you can see how there's normally just 2 chromosomes in each of these pairs. Well, for chromosome 21, there's an extra chromosome. So that is how Down syndrome is caused. So to get the karyotype of an unborn child, there is a process called amniocentesis. This is where a needle goes into the woman's stomach and takes out some of the amniotic fluid that's surrounding the fetus. And then genetic testing is done on that fluid. All right, so now let's talk about genes. We've heard lots about genes. So chromosomes are, remember, big, long DNA chains, right? It's just a whole bunch of DNA that's coiled up and into smaller compact units. But if you uncoil the DNA is actually made up or coding for genes. Okay, so a certain section of DNA here will code for a gene. And let's say this gene looks like this. We'll talk more about the DNA structure and those letters later. But the gene might code for an eye color, right? So it might be for brown eyes, or hair color, like blonde or brown hair color. So genes are actually coding for specific traits. Okay, that's what a gene does. So or 2 examples that we have here, eye color and hair color. But humans have over 25,000 genes in their DNA. So there are a lot of different traits that we can look at that are coded by our DNA. Now, what genetics is, what this whole unit is about, is the study of how these genes work to determine what characteristics get passed on from parent to offspring, and what's going to actually be shown. So some of these genes are actually coding for a protein. Okay, so there's a lot of different kinds of proteins. Down below here are some examples of the different types of proteins and what they could be used for. So there are proteins that are enzymes, and these speed up reactions in your body. So one might be like a digestive protein, like amylase, which helps digest your food. You have structural genes like keratin, which make up your hair and nails. Genes that code for proteins that are involved in transportation, so getting things in and out of your cell membrane. You have proteins that are hormones like insulin that regulate blood sugar. You have contractile proteins that allow muscles to contract, defensive like antibodies, which you learned about in the last unit. And you have some proteins that are for energy. All right, so we can't pass on these genes if we don't do cell division and replicate. So our cells are going to divide for 2 reasons. They're constantly dividing because we need to grow and we need to repair, right? So you get a cut. If you're young, you're growing older, your skin is constantly shedding cells. So that's one of the reasons our cells are going to divide. And when they do that, they're going through a process of mitosis. Okay. The other reason cells divide is to form gametes. And gametes are your sperm and your egg cells. Sperm and egg. Okay. And that process to form those is meiosis. So these 2 processes are slightly different because they're for different functions. When we're doing mitosis, we talked about it's for growth and maintenance. So we want the cells that we are reproducing to be identical to the originals. So we say 2N or diploid cells. You don't have to worry about that terminology in Science 30. But they have the same number of chromosomes as the parents. Okay. And this occurs everywhere in the human body. Now, meiosis, this is specifically for gamete formation, sperm and egg cells. And when you are creating a sperm or an egg cell, you don't want the full copy of all the chromosomes in your body. You want half a copy. So we are called haploid. So we are going from a full complement or a diploid complement of all of your chromosomes to haploid cells. And these are all your gametes or eggs or your sperm. So one cell here divides into 4. And there's 2 stages of division. 2 divisions occur there. Now, this process is only happening in the gonads, the ovaries, and the testes. Okay, and this is where you're going to get genetic variation. This is the cause for why we all look different and why we aren't just like identical copies of each other. We're going to take a closer look at both of these types of cell division. First off, mitosis. Remember, this is the one that is used everywhere in your body. You want to produce 2 identical daughter cells, we call them. So you might have this happening in your skin, hairs, as your nails grow, your hair grows, etc. So the major purpose, again, is for growth and to replace those worn out or dead cells. Okay, so for mitosis, a copy of the DNA must be made before replication. So here is your copy of each chromosome that's occurring. So your red one forms a copy, which are attached at the centromere, and your light blue one here, chromosome, forms a copy that's attached at the centromere. Okay. The chromosomes, we're doing this as though there's only 2 chromosomes. So this is like an ant cell, right? Remember, ants only have 2 chromosomes. We would have 23 of these in our cells. So a little bit more messy. So we're just dealing with ants. So the chromosomes line up at the center of the cell, and each one is pulled away from its partner. So you end up still with 1 copy of each of the chromosomes. So they're still considered diploid cells. Alrighty, so they're identical to the parent. You'll see identical to those parents, and that's what you want. You don't want every skin cell to be different. You want them to be identical to the one that created it. So that's mitosis. Now let's dig into meiosis. Meiosis is not creating identical cells. We are creating gametes. These are the male sperm and the egg. All right, so it's going to start the same way as mitosis. You're going to produce a copy. You're going to make a copy of each chromosome, and they're attached at the centromere again, and they're going to line up. Now, this is where some crossing over can occur. So when they line up, sometimes these chromosomes will cross and kind of switch genetic material. Then stuff gets all mixed up. All right, so then we do a division here. And then you'll see there's a second division. All right, now this leaves your daughter cells. Remember, they're all haploid, or they only have half haploid, half the DNA as the parent cell. This is because this, let's call it an egg cell, is going to combine with a sperm cell, which has another half of the chromosomes, and then together they're going to bring all the complement of chromosomes together so that you have a diploid cell. But you don't want to start with a diploid, or you'll end up with a diploid and a diploid, and you'll have way too many chromosomes in a cell. Okay, so the key parts to this in Science 30 that you need to know is that meiosis is for gametes, they form, which are sperm and egg, and the sperm and egg cells have half the number of chromosomes as a regular body cell. That is the key. As long as you understand that, then you are fine. You don't need to know interphase. You don't need to know any of that terminology. Okay. All right. So on the last page, I talked about a little bit about fertilization really briefly. Fertilization, this is where you have your sperm and you have your egg. They come together and you have a zygote. All right. And you'll notice there is one chromosome here from the sperm, one chromosome from the egg, and 2 chromosomes in the zygote or the fertilized egg. And every time this cell now divides, it's going to go through mitosis. And we saw when you go through mitosis, you create the same numbers of chromosomes in your cell. So every cell after this point is going to be have 2 chromosomes, it looks like, for this organism. All right, so fertilization. Right. Now, this is where you get the uniqueness happening, right? So this sperm cell, the chromosomes in there are coming from the father, the chromosome in the egg are coming from the mother, and the combination of these chromosomes together is what gives the uniqueness and the genetic uniqueness to each of us. So the mixing of our genes happens at this point. Okay. Although there's talking about here down below in this picture here, you do get some genetic similarity, as you probably noticed. You might have like similar colored eyes to your mother, or some kind of genetic trait from your father. And sometimes they can be super obvious, like this example of the Sheen family down below here. Martin and Charlie, I can't even tell them apart anymore because I knew them when they were younger and they looked exactly like him. And anyway, so you just get the idea here of you definitely are maintaining some genetic similarity, but you're also creating differences, right? So some of these brothers look quite different than some of the other brothers. So on this page, we have a little summary of cell division. I would recommend the video if you're still a little confused by mitosis and meiosis. It's a nice little video. Here is a good chart overview of just about some of the key things you probably want to know for mitosis. Remember, this is the one that's creating genetically identical cells or diploid cells. It's for growth, maintenance, repair. And there is no crossover going to happen here. We want to create identical cells. We don't want variation occurring, right? And one cell is going to create 2 daughter cells. From one cell, you're getting 2 daughter cells to grow. Whereas in meiosis, one cell is going to give you 4 gamete cells. So one cell is turning into 4 gamete cells. But this is going to take 2 separate divisions for that to happen. There's 2 steps to that process. All of the daughter cells here are half or haploid cells, so they have half the complement of chromosomes, and these are specifically for sexual reproduction. All right, so some organisms will reproduce asexually, make a copy of themselves, like bacteria can do that. So bacteria might use mitosis to make a copy of themselves just to asexually reproduce. But when they do that, they're not getting any variation. Sexual reproduction is where you get the variation from the crossing over and the mixing of 2 organisms' chromosomes. Okay, so that's your cell division summary. So you have a little bit of the background of what's going on when we're talking about genetics and passing on genetic material. All right, so another topic, terminology that you want to become familiar with is selective breeding. And we've probably all seen this somewhere, but maybe never really looked at the term before. You've seen it probably in your life if you have a certain breed of dog or a cat. Selective breeding is when we choose individuals with useful characteristics to produce a more desirable plant or animal stock in this case. So this example here for cattle, I should say. And this is for raising cattle for beef production. And it's looking at getting females that have a strong maternal instinct. That means they're going to nurture and feed their calves better, and the calves are going to survive better than females that don't have this nurturing instinct. Okay. They also want cows that have a high growth rate, right? So they're able to put on tissue, put on meat onto their body quickly. So you're going to look at two parents here. You have a mother with a strong maternal instinct and a father with a fast growth rate, and they are going to reproduce them. And here are some of the possible offspring down below. So here's an average maternal instinct with high growth rate. But what you want to find the strong maternal instinct and the high growth rate offspring. So this offspring, you like this offspring, so you're going to want to select this cow to breed and continue breeding. So you'll see a lot of this play out with dog breeders as well. They'll look for the dogs that have none of the traits that they want to pass on, and they'll select those dogs to be their breeding dogs. So here's another example of selective breeding that has occurred over about 7,000 years. Corn used to look like this, right? So this is teosinte. So it's the grass form of corn way back when. And over thousands of generations, the First Nations selectively bred corn to eventually become more like the corn that you see these days. So slowly and slowly, they found the corns that they liked, and they would plant those seeds and continue to select and choose the offspring of the corn that had the bigger kernels and these rows of exposed kernels that they could pull out of the cobs. So how did people come up with this idea of inheritance and the idea that there was genes? It was actually known for quite a while about this concept of genes, even before we could even see them. So in the 1600s, 1700s, there were some simple microscopes that allowed people to see cells like sperm cells and egg cells, but they hadn't still seen chromosomes yet, so they couldn't see that. Until Gregor Mendel came along. He was a monk, and he was one of the first people to start doing a systematic study of the inheritance of characteristics. So how our characteristics are passed along from generation to generation. And he was a big gardener, and he used pea plants, and he kept records of all the different generations for a long period of time. So this is in the 1860s. So one thing he noticed over here on the side here, so he had these parent plants. So one had purple flowers and one had white flowers. When you see this cross, that means he bred them. So he crossed them, or he bred them. And this crossing produced offspring, so seeds. And if you planted those seeds, that offspring, all the plants of that crossing of those parents all had purple flowers. Every single one of them. This is called the first generation. So you can think of them as the kids, but when we're talking about flowers, we don't say kids, we say offspring or first generation. Okay. So then he took the seeds from that generation and crossed them with each other. Okay. So they both had purple flowers still, right? So this is the same. So he's taking that same first generation, or the offspring, and he noticed with those seeds, 3/4 of them had purple flowers and 1/4 had white flowers. So he was like, what's going on here? So somehow this white gene, or he called them factor, must have been carried through the offspring but not shown until the second generation later. They were visible then. So this really piqued his interest, and he did a lot of experimenting to see what was happening to these factors. So not only did Mendel look at flower colors, but he also he looked at the pea pods too. So he did some experiments on pea pods that had round peas and wrinkled peas and crossed those. And so this is where that term factors, he used these factor of round peas and wrinkled peas. Nowadays we call these genes. So the round pea gene or the wrinkled peas gene. And he noticed when he crossed these round peas with the wrinkled peas, all the peas were round. Okay. Then when he took these round pea first generation again and crossed them, you guessed it, the same thing happened with the purple and the white flowers. The dominance number of the offspring were round peas. So about 75% of all the offspring in the second generation had round peas, but 25% of the offspring had the wrinkled peas. So somehow that wrinkled pea gene or factor had made its way again through that first generation. That none of them were wrinkled, but somehow they carried that wrinkled gene all the way through. So he thought that some genes must be more powerful than others. So in this case, that round pea gene must be more powerful. And if you have it and the wrinkled pea gene, you're only going to see the round peas. Okay, because they're masking or hiding that wrinkled pea. And this idea of dominance and recessive genes is what comes from his experimenting, and we will talk more about that in future lessons. So this just gives you kind of wet your whistle, gets you a little interested in genetics, and then we're going to dig deeper into a few more of these topics in the next lesson. Have a great day, everybody.